Beta-adrenergic-receptor kinase, also referred to as beta-arc, is a serine, threonine intracellular kinase. It is activated by pKa and its target is the beta-adrenergic receptor. It is one method by which the cell will desensitize itself from epinephrine overstimulation. Activation. Steps 1. Upon stimulation of the beta-adrenergic receptor by epinephrine, Gs will be activated. Steps 2 and 3. Gs alpha will then stimulate adenyl cyclase to make CAMP. Steps 3 through 6. CAMP will then activate CAMP-dependent kinase, pKa, which, among other proteins that it acts on, will phosphorylate serine and threonine residues on beta-arc. Step 7. Beta-arc, itself a serine, threonine kinase, will then phosphorylate serine and threonine residues on the beta-adrenergic receptor itself. Step 8. This will facilitate beta-arrestins binding to the receptor. Additional stimulation by epinephrine will now be unable to activate Gs due to arrestin. Therefore, beta-arc is a negative feedback enzyme that will prevent overstimulation of the beta-adrenergic receptor. Other similar systems. In the rhodopsin system, which regulates rod cell function in the retina, rhodopsin kinase will phosphorylate serine and threonine residues on the rhodopsin receptor. Similarly to the beta-arc system, the phosphorylated rhodopsin residues will then bind to arrestin, resulting in receptor desensitization. Structure. Protein structure. The structure of beta-ark1 consists of a protein of 689 amino acids, 79.7 kilodaltons, with a protein kinase catalytic domain that bears greatest sequence similarity to protein kinase C, and the cyclic adenosine monophosphate. Cyclic AMP, dependent protein kinase. Gene structure. The gene spans approximately 23 kilobases and is composed of 21 exons interrupted by 20 introns. Exon sizes range from 52 bases, exon 7, to over 1200 bases. Exon 21. Intron sizes from 68 bases. Intron L to 10.8 kilobases. Intron A. The splice sites for donor and acceptor were in agreement with the canonical GT-Ag rule. Functional regions of beta-arc are described with respect to the location within the exon intron. Organization of the gene. Primer extension and nase protection assays suggest a major transcription start site, approximately 246 bases upstream of the start ATG. Sequence analysis of the 5-flanking promoter region reveals many features characteristic of mammalian housekeeping genes, i.e., the lack of a Tata box, an absent and non-standard positioned CAAT box, high GC content, and the presence of SPE1 binding sites. The extraordinarily high GC content of the 5-flanking region, greater than 80%, helps define this region as a CPG island that may be a principal regulator of beta-arc expression. Interactions. Beta-adrenergic receptor kinase has been shown to interact with, see also, G-protein. G-protein coupled receptor kinases. Kinase. Beta-adrenergic receptor kinase at the U.S. National Library of Medicine. Medical subject headings. MESH. Human ADRBK1 genome location and ADRBK1 gene details page in the UCSC genome browser.